Okay, so no doubt till now.
Yes, sir.
Okay, perfect. So before we start genetics, we today have to finish. I'm selecting pen. Okay, we today have to finish the last topic, which is about infertility, right? Okay. So as we already discussed, did we discuss something about infertility in the last class or nothing? I think we discussed something, just the basics. Am I correct?
Yes, sir.
Okay, so what did we discuss? Can you quickly tell me? You can keep your NCRT open with you or any other notes that you like to refer so that you can answer and discuss things and we will understand things in detail. So what do you think is infertility? Or what did we discuss about it in the last class? Just tell me that. Or what is your opinion about infertility? Is it a problem? Is it a good thing? Am I audible, Rabia? Yeah.
Yes, sir.
Mhm.
Yes, sir. Infertility is a problem.
It's a problem, right?
Yes.
So what happens in this? The word infertile. What do you think it means? What do you think is the problem here?
Maybe the
It is so it has something to do with reproduction, right? It's a kind of inability. It's a problem because it's an inability. Remember in the chapter we also discussed about contraceptions. Contraceptions are the methods, ways, or drugs through which one can avoid pregnancy, right? One can avoid implantation or fertilization or fusion of gametes. But that is deliberate. That is when someone really wants to avoid it because they do not want unplanned pregnancies to happen. Make sense, right?
yes, sir.
So that is deliberate attempt. In infertility, it is inability. Like even if someone wants to have progeny, to have a kid, they cannot. Now that's why it's a problem, right? So what do you think can go wrong? What all things can lead to infertility in humans? I'm asking humans, but it basically holds true for all organisms when we say infertility. But since we are more concerned about human here, so tell me, what do you think can lead to infertility in humans? What can go wrong?
Diseases, any diseases.
Diseases, right? So first, let's define it in a way. So it is inability to produce offsprings, right? Inability to conceive pregnancy. Infertility is used can happen to both males and females. And when we talk about the causes, you are right. Certain causes, it could be either due to diseases. What sorts of diseases do you think, Rabia, can cause infertility? What do you think can cause infertility? What kind of disease? If you don't know the exact name of the disease, that's completely fine. You don't have to tell me the name. You have to tell me what kind of can cancer cause
Hello, sir. Your voice is not visible.
what do you think, Rabia?
So your voice is
Yes, Mariam,
lagging.
how are you? Is it still lagging? Oh no, my internet connection is unstable, it says. Is it still lagging?
No, sir, it's fine now.
It's fine. Okay, perfect. So my internet connection went weak for some time. Is it now okay?
Yes, sir.
Okay, perfect. So I was asking Rabia that what kind of diseases do you think can cause infertility?
Uh, sir, PCOS or PCOD.
Mostly those diseases that can cause affect either the gonads, right, or reproductive organs, correct? So it could be a RTI that is affecting the uterus, that is affecting in males. It could be some disease that affects the penis and the testes. It could be PCOD or PCOS, as you said, which affects the ovary and its ability to form ovum. Correct? It could be problems with the uterus, a cyst in the uterus, a tumor in the uterus. It could also be the problem with the birth canal, cervix, vaginal canal. All of it, which makes it very difficult or nearly impossible for an organ and a human to reproduce. Make sense? Yes, no?
Yes, sir.
Very well. Perfect. What could be other causes apart from diseases? Could it just be diseases? Anything else that can cause infertility in humans? What do you think can cause?
Uh, drug.
Yes, it could be overuse of drugs, or it could be radiation, right? It could also be why radiation? So drugs and radiations, because they affect the gametes. If someone abuses drugs, alcohol, or any kind of like over limit, or lives in a high radiation zone where radiations can affect the sperm count of a male, then they are more susceptible to infertility. Third, but a little rare factor could be genetic. Like infertility could be genetic because of some mutation somewhere that leads to not formation, like less formation or not forming gametes at all. So it could be genetic as well. Okay. If an infertility is by birth or if it is genetic, we call it sterility. Okay, if the reason is genetic, we call it sterility, and the organism is called sterile. And you know about one such way that it can happen. If we make crossbreeds between two different species, like if you make a crossbreed between a horse and a donkey, the offspring of that crossbreed is called mule, and you know that mule is sterile by birth, so it cannot go on to give rise to offsprings. Make sense?
Yes, sir.
Yeah. Okay, very well. Now, whatever be the reasons that causes infertility, we have Iram also joining in. How are you, Iram and Mariam?
Good, sir. How are you?
Yeah, good, perfect, great. I hope you're doing good. We just finished discussion on infertility, and we will just take 10-15 more minutes to wrap up this thing before we go to genetics today. And just to let you know, and let me check. Yes, so the spotlight thing is on, just in case if you want to switch on your videos, which I will highly appreciate. Okay, so whatever is the cause for infertility, it's not a good thing, because humans do not want to not be able to have progenies, though having more progenies is also a problem. So for that we have contraception. So for that we have methods, planning commissions, and in the last class we discussed the role of providing sex education early to adolescents so that they can plan their lives better in terms of their sexual life better and their reproductive life better, and prevent themselves from diseases, etc. So that is a different thing. Now we also have to find solutions for infertility. Now for infertility, the solution that comes into picture is called assisted reproductive techniques. Okay, and its acronym is ART. So these techniques basically are based on the logic that what can lead to infertility. So in infertility, as Rabia currently pointed out, whether it is diseases, drugs, or genetic, one thing that happens in most of the cases is that either one partner is not able to produce good gametes, or other thing that happens is, mostly in females, it happens is that the reproductive tract is not capable of holding pregnancy, or it's not very favorable for implantation to happen. Okay, so both these things are tackled in slightly different way. Let's first go to the problems where gametes are an issue. So what do you think, if gametes are an issue, what can be done? When I say gamete is an issue, I mean that they're not forming gametes, there are not enough gametes, or gametes are not fusing, fertilizing on their own in a natural condition inside a female's body. So what should be done, or what can be done? What do you think? So first write down what's ART, what is assisted reproductive technique, write down. Thank you so much, Iram, good to see you. And you can write that these are methods by which human couples can be assisted. These are methods by which human couples can be assisted to have children using special techniques. To have children using special technique. Okay, now one of the most common problem that happens is that males are not able to make enough number of gametes. Remember, I told you that in per ejaculation, there should be around 30 million sperms on an average, 30 to 300 million sperms on an average. If a male is not forming that number of sperms, then the chances of fertilization drops severely. And that kind of problem is called oligospermia. So one issue that happens in males is oligospermia. The word oligo means few or less, and oligospermia means not able to produce enough sperms. So less sperm production. Now, oligospermia can have multiple reasons in males. Lifestyle, abuse of drugs, radiations are major reasons. And in case of oligospermia, so males cannot produce enough sperms to fertilize the ovum. Everything in the female system, let's say it's normal. So what can you do? You must have heard of a term called test tube baby, right? It's a very popular term. That's also a misnomer. So you cannot develop a baby in test tube. And at any given time, I don't know if any test tube is ever used in this process. I don't know why they named it test tube baby. So the fertilization is, in this case, the scientific name for this technique is called IVF. Fertilization. Your meaning in vitro, people? What does in vitro means? You know that? So the word in vitro means outside the organism's body or outside the organism and inside some glass vessels or something like. So when you do any reaction in your chemistry lab, you are doing all the reactions you do are in vitro reactions. You do it in a flask or a beaker or a test tube, something like that. So that's called in vitro. So in this technique, male and female gamete is taken from their body. And in this case, we don't need millions of male gametes. We can even work with a few. So basically, artificially, you keep one female gamete, which is ovum, and you can keep a few male gametes and allow them to reproduce without any hostility, without any selection in a Petri plate or in any vessel outside the body. But you must be thinking, or you can imagine the level of sterilization, the level of cleanliness that you need, and the level of sophistication that you need so that this process is successful, right? The gametes themselves should not get infected. The gametes themselves should not die. The gametes themselves should not mutate or become something else. So it's a very, very sophisticated technique in which a male and a female gamete is allowed to fertilize. Write down. Male and a female gamete is allowed to fertilize outside the female body, outside the female body, by creating similar environment, or similar conditions as that of the female body, Now, you can now understand that it is very important, like temperature should not be high or low. You cannot mess up with temperature, you cannot mess up with fluid. So when you take male and female gamete, human male and human female gamete, you cannot just keep them in water because they will die. So you need pH, you need salinity, you need viscosity as same as it is inside the body. So when you achieve that, then you allow this fertilization to happen outside the body, and that's called in vitro fertilization. But just by doing in vitro fertilization outside the body, the problem is not solved, right? So you solve the problem that fertilization was not happening, but when you took it out and let's say fertilization now happened, then what? Is it a solution? As I already told you, you cannot develop a baby in a test tube or a beaker or a flask, right? Baby can only develop as of now with the current techniques and technologies that we have only inside the mother's womb, right? So what they show you in science fiction movies like Matrix, that inside a pod or inside a chamber, a human is developing. That's the concept of artificial womb. Now, science as of now is a little far away from it, but it's not impossible. It is the future. In future, people will be making human babies outside the human body, just like fruits are ripened on the trees. You can, but it's far-fetched. It's a science fiction for now. Every science fiction is a reality, science reality of the future, right? 400 years ago, flying with the aeroplane was a science fiction, but now we do that. 200 years ago, you taking these classes, you talking to me and seeing me from from thousands of kilometers was a science fiction, but it's not now, right? We all can see each other. Maybe we will be able to 3D, 3D imaging, you already know, you can just make any person come anywhere as a 3D impression. This is also possible. So artificial womb is also possible, but for now, let's stick that it's not happening. So IVF does not solve the process. You have to, okay. You have to transfer the gamete after IVF into the female body back, right? Now it will become a zygote. After fertilization, it becomes zygote, and then it develops into embryo. So write down, IVF is always followed by IVF is always followed by transfer of zygote or embryo, transfer of zygote or embryo inside the female reproductive tract. Inside the female reproductive tract. Now, so transfer is very important. Now, if you are doing, so what is the difference between a zygote and an embryo? Zygote is a single
Zygote,
cell. Yes,
yeah.
yes, Rabia, tell me.
Yes, the zygote is a single cell, and embryo is made of many different cells.
So embryo is multicellular. A zygote is
Yes.
only called zygote till it is one cell stage. So two things fertilize, it becomes one cell, and it's called zygote. The moment it divides, which in humans happens after 24 hours. So for 24 hours, it stays a zygote, and after it divides and becomes a embryo. Make sense? So
Yes, sir.
let's say, so if you are just after fertilization, you are putting back zygote. Where should you put it in female body? Now this is the logic, so that you don't have to mug up any fact. Think logically. Where was fertilization happening naturally? What is the site of fertilization? Iram, you are mute.
Oviduct.
Oviduct, right? Ampulla isthmus junction, to be very precise. Correct? So that's where fertilization was happening. So where do you think zygote spends its time before it becomes embryo inside a female? If fertilization is happening in ampulla isthmus junction, zygote, most of the zygote's stage time will go in the fallopian tube only. It is still moving in fallopian tube to come into the uterus to get implanted. And I already shown you a figure where first the zygote becomes two-celled. From in the 2 to 8 cell stage, it's called a morula. Then it becomes a blastula around 16 to 32 cell stage. Then it becomes a blastocyst, and then it gets implanted in the uterus, right? So if I have to put zygote back in the female body, where should I put it?
fallopian tube left.
Perfect. So we have to put a zygote back into fallopian tube, and that technique is called ZIFT. And the logic is ZIFT stands for zygote intrafallopian transfer. Make sense? If you are transferring zygote, you have to transfer it into fallopian tube. So it's called ZIFT. Okay. Now, if you are not transferring zygote and you are allowing that after fertilization, you want to make sure that the fertilization is normal, and you want to make sure that for some cycles of division, it should divide properly, and you can see it under the, like you can see it for yourself. Then you allow zygote to become embryo, and then you transfer the embryo. Then where will you transfer embryo? So if you're not doing zygote transfer, then you could do embryo transfer. It's called ET. And where should you transfer the embryos? In the same fallopian tube. Can I transfer embryo in the fallopian tube? Yes, no, what?
In the endometrium?
Yes, in the uterus you mean, right?
Yeah.
Perfect. So when we do embryo transfer, we often do it when the embryo is more than 8 blastomeres. Now it's already a blastula, right? If it is more than 8 blastomeres, it is a blastula, and that is called IUT, intrauterine transfer. Makes sense? Why? Because now it will rapidly divide to become a blastocyst and will embed into the walls of the uterus. Is it clear?
Yes, sir.
Now there is one more thing where, so this is called IVF, which is followed by either ZIFT or ET, IUT. Okay. Now sometimes there is a concept of donor females. So what happens is, the problem is, so this is done mostly when the problem is with male not being able to form gamete and all, right? You can do one more thing if male cannot form much of gamete, which is called intrauterine gamete transfer or artificial insemination. So another thing is artificial insemination, and this is done in cases where males are either not able to produce enough sperms, or males have erectile dysfunction. Now erectile dysfunction, so their penile tissues won't function properly to transfer the gametes into the female tract. So then females, the couple can go for a artificial insemination where males, from males, gametes are collected, their sperms are collected, and then they are artificially introduced into the female cervix. Make sense? Okay, so that's called
Yes, sir.
artificial insemination, AI. I'm writing these short forms or acronyms because sometimes they ask you this. What the, so what they can, the question can be like in an entrance, one of the question was, they will give you a case study, something like that, that a couple went for IVF. So you should know what is IVF, which is in vitro fertilization. And then transfer of zygote happened. So what is this technique called? So IVF and then transfer of zygote is called ZIFT, zygote intrafallopian transfer. And if they underwent embryo transfer, it's called IUT, intrauterine transfer. Okay. or ET. Artificial insemination is AI. You can also do so artificial insemination is done through, as I told you, intrauterine insemination or transfer. It's called, sorry, intrauterine transfer is for the embryo, but IUI, intrauterine insemination, this is another thing that we can do if the males are not able to form enough sperms. Is it clear?
Yes, sir.
Now, in some cases, the issue is in the female system. Now, some females are not able to produce and release proper ovum. Okay, in that case, regardless how many sperms male send into the system, there will be no fertilization and there will never be a pregnancy. Now, if that is the case, there are two ways to solve it. One way is, can you think of the ways that can solve this problem? Female is not able to produce or secrete good gamete or a viable ovum. Then what should be done? What can be done? In that case, you can go for, so it's not like you cannot take ovum from the ovaries. Can you tell me why? We cannot just put injection in the ovary and take ovum from it. Ovums are mostly collected when they are after post ovulation on the 14th day, when they are in the fallopian tube or when they are in the uterus. Then only you can collect it. It's very difficult to collect it from ovary because in ovary multiple things are at, all the follicles are developing and one follicle will be released, right, every month. So we need the most mature one, we need the ovum. And the reason we cannot collect from ovary is because ovary does not have the most mature form. It secretes secondary oocyte, correct? So we only can take it after secretion. We need secondary oocyte. So that's why we cannot take it from within the ovary. So what we can do is we can find a donor female. A female who's normal, who's producing normal ovums and releasing it every month. So in this case, a female will donate her gamete or her ovum, and that ovum will be taken and transferred into the recipient female's fallopian tube. Okay, just by with the injection. And then that female can go on and the male can transfer gametes normally because everything else is normal. Male is forming normal number of gametes. So male can naturally inseminate and it will lead to pregnancy. Make sense? Yeah. So that's
Yes,
called,
sir.
the acronym is very fitting. That's called gift. One female is giving a gift to another female, and that gift is the ovum. And it stands for gamete intrafallopian transfer. Because you are not transferring an embryo or a zygote, you are transferring a gamete only. Gamete intrafallopian transfer. We never transfer one sperm into the fallopian tube. You know, that makes no sense. We don't know where the ovum is at what time point to transfer a sperm. And if you have to transfer sperm, just transfer a million of sperms in the cervix. No need to go into the fallopian tube and doing that thing. Here also, but ovum you can transfer in the fallopian tube because that's a big cell, and the female system will not kill an ovum, right? But it will kill sperms. The ovum might still die because of other reasons, and that's why gifts is not very successful in the first attempt. You have to do multiple attempts of that. But you understand what is gift? Write down where the ovum is collected from a donor female and transferred into the fallopian tube of a recipient female or another female who cannot produce it herself. Okay. But in this case, the doctors have to be sure that she cannot produce ovum, but she can hold pregnancy. The uterus or the womb can provide all the suitable environment for the pregnancy to happen. In some females, and this part is not, I think it's not covered in your NCRT, but I'm just telling you, in some females, the problem could be exactly opposite of it. They do produce ovum, but they cannot hold pregnancy. So the problem is not in the ovary, the problem is not in the fallopian tube, the problem is with the uterus. What do you think can be done in that case? So if a female is not making gametes, you can take a gamete from a donor. If a male is not making enough gametes, you can collect the male's gamete and inseminate in the female system. If the male is not forming a lot of gametes or forming very few, you can just take male and female ovum and make them fertilize outside IVF and then transfer it back into the female body. But if a female body is able to produce gamete but is not able to hold pregnancy, the problem is in the uterus environment, then what can you do? Which, by the way, many celebrities go through. You know the answer. The problem is with the womb, so you hire a womb. What is called?
So there's a like
Surrogacy.
Yes, it's called surrogacy. So the female donates the ovum, who's the actual biological mother. The male donates the sperm, who's the actual biological father. It is fertilized outside IVF, and then you get a surrogate mother who's willing to host the pregnancy for. So biologically, the baby is not of the mother who's hosting the pregnancy. Biologically, the genes, so the baby will look either like the biological mother or like the biological father. The baby will not look like the host mother. Okay, so she's just hosting the pregnancy. In her womb, the baby will develop and get nutrition and produce wastes. And now this is a kind of a ethical debate around it, whether who should be the real mother, the one who just donated the ovum or the one who hosted the pregnancy for 9 months and in whose womb the baby developed. But mostly these kind of things are done on a legal contract based thing. So legally, before you start everything, the surrogate mother and the biological parents have to sign contracts and have to agree that after the birth of the baby will be like the biological parents will be the custodian and all, then only it can be done. So that's called surrogacy. It is also a assisted reproductive technique, but in cases where female bodies are not capable of holding pregnancy. Any doubt in any of these terms or any of these concepts, people? Yes, no?
No, sir.
No. Okay, perfect. So shall we start with the unit genetics and evolution?
so
Yes.
Yes,
so
Hiram.
there's one more thing called ICSI.
Oh, yes, right. So one more thing called IC. Yeah, sorry. So what Hiram is talking about is intracytoplasmic. What is it? ICSM, intracytoplasmic sperm
Sperm
injection.
ejection.
Yes, injection, right? So ICSM is in the worst of the scenarios where the father is not able to either producing very few sperms, or sometimes you have the father's sperm and the father is not available, you know, but still the mother wants to have a child of that particular male of the father. So you can cryopreserve your sperms and ovum, you know that. So if you know about a very famous celebrity, Priyanka Chopra, she froze her eggs, and she recently in an interview told so, and many people do that because at an early age, when you are around in your 20s, in your early 30s, for females, the quality of their eggs and the ovums is the best. As the females age, when they are in their late 30s and 40s, they cannot produce the same quality ovum, right? And you know, the quality of ovum is very important for the formation of the baby, because everything that the baby is getting, protein, RNA, it gets from the mother. From the father, it only gets the genetic material, right, which is in the head of the sperm. So you can freeze, biologically freeze your female can freeze her ovums, and then it can be later used for a IVF and surrogacy, or IVF and putting back into her own body, anything. So in those cases where the sperm is limited in number, we go for one thing called ICSI, right? It's called intracytoplasmic sperm injection. As the name suggests, what it means is you have a ovum. Remember, ovum has layers, protective layers on the top. So the actual cell of ovum is this. Then you take a very fine pipette and hold the ovum in place with some pressure. So the ovum is holded in place. And then there is a very tiny needle. Okay, this very tiny injection needle, which we use. And it carries a sperm inside it. Okay. So you technically inject a sperm into the cytoplasm of the ovum. So it's called intracytoplasmic sperm injection. So, and imagine how much sophistication do you want to make a needle that tiny, which can penetrate the ovum. But you also should know that ovum is one of the biggest cells, as I told you. Ovum is a very big cell, right? It is the biggest cell that a human has throughout its life cycle. So it's not impossible, but this can be done. Okay, and this is called ICSI. Is it clear? You know, you can write down ICSI is injecting male gamete directly into the cytoplasm of the female gamete ovum. Thank you, Iram, for bringing this up. Just forgot about it. Is there anything that we are missing out? Can you please check the NCERT? Iram, is there anything else that we are missing out? No? Okay, perfect. So let me start a new page here for chapter 5. Anyone has any doubts? I'm giving you a 2-minute break here, people. It's exactly 53 at my clock. I'm giving you 2 minutes break to drink water. I'll also have a water break, to look at your notes of the previous chapter, and then we'll start with chapter 5, which is, what is the chapter name? Principles of Inheritance. Genetics and inheritance, right? Heredity and inheritance, something like that. What is it? Can anyone confirm?
Principle of inheritance and variation.
Sorry. Inheritance and variations. Yes. Genetics is one of the favorite topics of, personally, I like genetics a lot in biology. It is very, very logical a thing. When you study genetics, you realize that everything has a reason. Everything that life has a reason. And we can trace back every phenotype from your curly hair to your eyes color to your cravings to some extent also. Whether you are a sweet tooth or whether you like certain kind of food, other phenotypes, whether you are you cannot digest certain kind of food, you cannot tolerate heat, you cannot tolerate cold, all these things, the pattern of a male's beard, the texture of a woman's hair, skin, everything has a reason, and which is present in the genome. So remember, I asked you in the last class to go and figure out a few words. What were these words? First was right. Second was RNA. Before I go ahead, I will discuss these things, and then genes, and then chromosomes, right? Okay, so let's start with first. Who's going to talk about DNA quickly? Tell me, what is a DNA? Since you people have done this. What is a DNA, people? Anyone would like to attempt? Mariam, Rabia. Am I audible clearly, or is my voice breaking? That's why you're not. Is my voice breaking?
No, sir, you're audible. You're audible.
Okay, perfect. Yes. So tell me, what is a DNA? Have you heard this term before studying biology? I think this is your 4th year studying biology. So in these 3 years, have you heard this word anywhere?
Yes,
Of
sir.
course, in 11th class, you have even made the structure of it and dissected it to say that what is present inside and all things like that, right? Nucleotide, nucleoside, everything. But then you forgot. Gave the final exams, you forgot, right? Yes, tell me, Rabia.
It contains the genetic information.
DNA contains the genetic information. What is that genetic information? A letter from someone, a movie clip, song, a WhatsApp message, a tweet, Instagram upload. What is that information that DNA is holding in it itself? See, I always believe in simplifying science, right? Yes. Sorry, Mariam, come again. Sorry, I could not hear the few words of the beginning.
Cells that contains genetic information.
What it contains? Sorry.
So the molecules inside cells that contains the
molecules inside. Yes, molecules inside cells. She says there are millions of molecules inside cells. There are proteins. There are enzymes that work in the cell. There are structural molecules that make the cell, like actin, myosin, microtubules. There are lipids. There are carbohydrates, which are sugars, glucose that get metabolized in the cell. So which molecule you're talking about? We all study science, we don't simplify it. I don't know why. It's not that difficult. So if I ask you what is a DNA, and I ask you to explain a layman a DNA. For example, you have to explain your 5-year-old sibling. Let me tell you what is DNA. What are you going to tell? The molecules inside the cells, he will understand nothing at all. Or your parents, if they're not from science background, let's say your parents are working in some business sector or something. If your mother is working in some other sector which is not science, or if she's a housemaker, how will you explain her DNA? If your mother asks, if your father asks, what did you learn today in Rahul's class? Like, we did some complicated things, just important for boards, not important for us. So forget about it. Mariam did some. Yes, Mariam, would you like to simplify your effort? It shouldn't be that difficult, people. We all have DNA, right? Come on. As a biologist, we should know what is DNA. That is the first thing that anyone can ask you. How do you even believe that you have a DNA? I'm like, all these could be just made up stories, right? Some people just decided, okay, let's make a syllabus. Let's make some fancy terms. Have you ever seen a DNA, Mariam? Have you seen your DNA? Are you convinced that your body contains DNA?
Yes, sir.
Why are you
Yes.
convinced? Why are you convinced that your body contains DNA? Like, I was not convinced for a very long time that my body contains DNA. I'm like, whatever, they're just lying. They could be just lying, right? You never thought that what is the proof that my body contains DNA? Okay, tell me. Why are you convinced and what do you think is a DNA? Try, at least try, Mariam. I want you to try. Come on. You're doing great. You talked about something called genetic information. See, genetics become super easy when you understand what are we talking about. And genetics becomes super hard when you just try to remember it. You can't remember genetics. No one can remember genetics, not even the geneticists or the scientists who are geneticists. You have to just understand genetics. Okay. So tell me, why are you so convinced, Mariam, that there is a DNA inside your body? And where is it and what it is? Let's start with some help. Let's give some help to Mariam. Let's start with the name. What is DNA? What does DNA stands for, Mariam?
Yes, sir.
What does DNA stands for? What is it? What is the full form?
Sir, something, sir, I forgot, sir, the name.
No, not a problem. Yeah, I am also not a big fan of remembering things. That's where I'm telling you from now onwards, if someone asks you what is a DNA, you don't even have to give the full name of it. You can just explain without that. Yes, Rabia, you're saying something. You would like to help Mariam? You know, whenever you feel, always mark my words. I'm teaching from last 7 years now. I started teaching very early in my life, and I have realized just one thing. If a student does not know something that he or she should have known, it's not the student's fault. It's the teacher's fault. The teachers were crappy. Okay. And as a teacher, I'm saying this to you. So you have all the rights to let your teacher know. I did not understand that. Even if it is happening after 20th time, it's okay. You have to just say, I could not understand. It's not your job as a student to understand. It's my job as a teacher to make you understand. You understand what I'm saying?
Yes.
So there is nothing like a bad student. There is only a bad teacher. Believe me. Okay. So never feel inferior, ashamed, hesitant when you don't know anything. We all don't know everything. Okay. So it's completely fine. If you don't know it, maybe the teacher was not good enough to have your attention, to make it so interesting that you try to know it or you feel like knowing it. Maybe you were sleeping in your class. So when students sleep in a class, students are punished. I believe teachers should be punished. How can a student in your class sleep? Like if a student in my class sleeps, I will just tell the student to please go and sleep because you're sleep deprived. I will not admit that someone can just sleep in my class because I'm boring. I try my level best to not be boring. Okay, over these 7 years, one thing I've learned is never be boring while you are teaching because students will just doze off. Okay, so it's always bad teachers. Yes, Mariam. So it's not a problem if you don't remember the full form. Rabia or Iram can help. Can you tell me, Iram, Rabia, what it is called? DNA.
Deoxyribonucleic acid.
Oh, wait. Went like a super fast train. And deoxyribo, blah blah. What? Sorry, deoxyribo what?
Deoxyribonucleic acid.
Nucleic acid. Okay, that's perfect. So I have in caps D, N, and A. Correct? Deoxyribonucleic acid. Let's start for RNA also. What does RNA stands for?
Ribonucleic acid.
Ribonucleic acid. Okay, that sounds like some relative of DNA only, you know. It's like Rama Prasad Mukherjee and Shama Prasad Mukherjee. The very similar, right? It's deoxyribonucleic acid, like Mahesh Kumar, Ramesh Kumar, something like that. So are they related to each other? RNA and DNA? It seems like they are related. Okay. What are genes?
Genes are the segment of DNA.
Okay, that's interesting. So genes are nothing but DNA itself. That's what Iram is saying. Then what is a chromosome? So let me write few things here. These are very interesting things you're saying.
DNA is present in a chromosome.
DNA is present in a. So what is a chromosome? If you're saying that coins are present in a box, I'm talking about the box. Then I'm not interested in the coins. So what is chromosome? If DNA is present inside chromosome, you cannot define like that. So what is a chromosome? Genes are, you are saying it's DNA itself. So genes are a segment of DNA, which means if this is a full DNA, this pen is a full DNA. If I cut it into two halves, one half you can say it's a gene. Let's say for now, okay? We will go into the details. But what is chromosome? It is a box in which DNA is kept. Is it? Or is it a wire on which DNA is hanged? Or is it a liquid in which DNA is floating?
So I don't know the exact definition of chromosomes, but it's like,
don't worry about exact definition. So if DNA is like ruafza, then is chromosome like the water for it? Or if DNA is like the coin, is chromosome like a box for it? What?
yeah, I believe so. That's
You believe that chromosome is something in which DNA is kept. Okay, Mariam and Rabia, do you also believe something similar, or you have some other theories for it? What is a chromosome?
I said chromosome is found inside the nucleus.
Chromosome is bound inside the nucleus. Perfect, you are right. I think I also studied something like that in books. But what is chromosome? That doesn't answer it. I'm asking you, what is a car? You're like, car is what runs on the road. But what it is that is running on the road? Horse can also run on the road. So for that matter, if you say that chromosome is bound inside a nucleus, there are other things also inside a nucleus, not just chromosome. So what is chromosome is my question. Good attempt, Mariam. Keep making those attempts. Come on. Rabia have not spoken anything. I think Rabia will only speak the right answer. She is saving her words.
No, so it's not like that.
So what do you think is a chromosome, Rabia? Do you also believe with Iram that it's something like a box in which DNA is kept? Or do you believe?
Yes,
so.
it's like
Yes, Iram.
So I don't say it's a box, but it's
See,
more
Rabia
like
just agreed and Iram just changed her answer. So Rabia just agreed that she also believes that chromosome is like a box in which DNA is kept. And then Iram is like, I didn't say that. And that's how the world functions, Rabia. You know, when you support someone else's cause, they change the cause. And then you're like, oh, what was I supporting? That's funny. That's great. Yes, great. Yes, Iram, what did you say? If you did not say this.
so I'm saying that it's not a box, but more like
Huh?
threads.
Or like chromosome is like a thread. Then what is DNA?
DNA is a part of this thread, or maybe present in this thread.
Okay. So chromosome is a thread and DNA is present in the thread. Okay. So DNA itself is then the thread, right? No, I don't think what can be present inside a thread. A thread is a thread. So do you mean to say that genes are nothing but DNA and DNA is nothing but a chromosome? Are you saying that? Which means all of them are same. Gene is a DNA and DNA is a chromosome. How can we be confused about this after 3 years of studying biology? That tells us how badly sometimes biology education is designed. And I'm very amused and happy to know this. So let's fix it. Okay, Rabia, you have some final thoughts because you supported Iram and Iram just changed her cause. So what do you believe now? Do you still believe that DNA chromosome is like a box in which DNA is kept? I just don't want to give you a definition and move ahead, because next year if someone asks you the same, you will just be same, confused in the same manner. These all words are important. Okay, I'll go for a final word from Mariam on chromosome, and then we'll move ahead. Mariam, do you have any final wise words about chromosomes? You can say that chromosome is like a envelope in which a letter is kept. Is it like that? Okay, no words of wisdom from Mariam, I think. Okay, so let's move forward. Let's start with DNA. So DNA, deoxyribonucleic acid, as you know, it's an acid. It's an acidic molecule. Okay, so this nucleic acid is a molecule itself. So DNA is a molecule itself, and a molecule is a group of atoms, right? And you have studied in class 11th, I think to some extent, not in detail, what are these atoms that make a DNA, right? You know about nitrogenous bases, right, that make DNA, correct? There are 4 types of nitrogenous bases: adenine, guanine, cytosine, thymine. They are called ATGC. That also makes the sequence of DNA. So DNA is nothing but a molecule, a very long molecule. In fact, the longest molecule which is present in the any organisms, any living organism system. Now DNA is so long, and DNA is double helical, double wired. So 2 pairs come together, like for a DNA. DNA is a very long molecule that looks like this, but not just 1 strand, but 2 strands. Okay. And these 2 strands are held together by nitrogenous bases forming bonds inside. This image you must have seen somewhere, right? Has anyone seen this image in reality? Who? You have seen this image, in reality? Anywhere? In by reality, I don't mean that in videos and in the textbook. That's not reality. That's what virtual virtual is. Reality means you seeing it under the microscope or with your own eyes inside a cell. Actually, no one has seen this thing in reality. The structure is deduced logically. We will talk about it how. Now, what is RNA? RNA is just a single stranded. There are double stranded RNAs as well, but we will not talk about them today. So RNA is derived from DNA. Okay, so this is DNA. And from DNA, if you can make a single stranded copy, that's called a RNA, where the bases are open, not tagged to anything. This is a RNA. So it is single stranded. Now, this is double stranded because of one missing oxygen. Deoxy, deforestation, demotion. You remove it, right? So deoxy means there is one oxygen missing from the structure, and that oxygen makes it stable and allows it to form double bonds. So one strand can form bond with another strand, and it is a more stable structure. So DNA is more stable than RNA. Just like a ladder. A ladder has 2 backbones, right? That's why it is stable. If it has just one backbone, it is not very stable. It can break easily. And on top of it, it is helical. It is coiled like this. It is coiled like this to make it even more stable. Now, DNA contains genetic information that Mariam said as base pairs, as bases. So let's say this first base is adenine, second is cytosine, third is guanine, fourth is thymine. Okay, fifth is again guanine, sixth is adenine, things like that. So from starting till end, there will be a sequence of bases that is present inside the DNA molecule. Okay, and this is the information that we were talking about. So this sequence of base pairs is the genetic information. Okay. Now, ribonucleic acid also carries the same information. There's only one difference, that RNA contains uracil, which is U instead of thymine, which is T. So RNA, if let's say this RNA came from the same DNA, so it will read as A, C, G, U, G, A. Instead of this T, it will have U. Okay. Uracil instead of thymine is the difference, and it is single stranded, unlike DNA. DNA is double stranded. Everything is clear till here? Makes sense? Mariam, Iram, and Rabia.
Yes, sir.
So if someone asks you now, if your parents ask you, what is this DNA thing that you were talking about in the class? So you can tell that DNA is a very long molecule which is present inside each and every cell of our body. Okay. Now it is so long, then how the cell stores it? So I always find it very difficult to store my earphones, the wired earphones, or any wire that I have, like my laptop charger's wire and things like that. It's very difficult to store things which are thread-like and very long, right? Your mother's always stores threads in home, right? So how do we store threads? In general, in life, how do we store threads? Do we just bring it together, make a clumsy thing and put it somewhere in the box? Next time when you try to open it, you can't. It will all be messed up. My earphones have this special ability to just get entangled on their own. Half of my life is detangling my earphones, so I shifted to Bluetooth. I'm like, life sorted. So imagine about a cell. A cell cannot shift on a Bluetooth DNA. It has to deal with this thread. So how should we deal with things which are very long and we have to store them? Simple logic. Anyone? Mariam? Rabia? Yes, Iram. Iram is like, I know the answer. I've been storing a lot of threads already. Tell me, how do we store threads? Very long things.
And just compactly roll it.
Exactly. You compact it, but in a pattern, which you can again go backwards and unroll it. Rolling a thread in the form of a spool of, right, is how our mothers have all these different colored threads in a box. So if I open these threads could be just is my voice lag? Is my voice lagging? Am I audible?
Yes,
Is
sir.
my voice lagging, people?
Yes, sir, it's lagging.
Is it still lagging? Let's wait for some time. I've turned off my video, if that helps. Is it stable now, people? Am I audible to all of you?
Yes, sir.
Mariam and Iram?
Yes, sir.
Yes, sir.
Perfect. So let's work with audio for some time. Okay, maybe my network is weak. So the one good way to keep any long thread-like structure is to roll it, or in a pattern, in a very neat and clean manner. That's called compaction of a long thread. Similarly, inside a cell, this very long molecule, which it could be like meters long in a single cell, and there are not just one molecule is there. So there are, how many molecules of DNA are present in one human cell? Anyone knows the answer? Okay, no. Now you will
One.
know the answer. Yes, Iram. How many?
One.
Okay, great. So that is what I was expecting. So that's not the right answer. So you will tell me the right answer in just 2-3 minutes. So as I said, there are multiple long DNA molecules present inside each and every cell of our body. So for, let's say, one is a green colored thread, one is a red colored thread, one is a blue colored thread. So we make spool of each of this. When we compact a thread, we need something on which we can compact it, right? Or roll it. We need a base, yes or no? We can't make a spool of thread just by thread. We need something, either a cardboard base or a pencil or a stick or something on which we can roll it. Yes or no? Make sense? Do you understand what I'm saying, or I'm just?
Yes, sir.
Yeah. So that base in our cell is proteins. So we roll this long DNA on proteins. We coil them over proteins. And these protein plus DNA coiled together is called a chromosome. Now, if I ask you, so chromosome is nothing but a compact form of DNA, which is compacted with the help of proteins. So these proteins are called DNA binding proteins. They bind to the DNA, coil it around, compact its length, make it small so that it can fit inside the nucleus. So DNA inside our body stays in the form of chromosomes. Now my question, coming back to the question. So DNA is nothing, chromosome is nothing but a coiled form of DNA with the help of proteins. So you can write chromosome is coiled DNA along with DNA binding proteins. And you also know the name of these proteins. These are called histone proteins. Histone proteins. Okay. So DNA wraps around histone proteins and becomes a chromosome. There are other proteins as well that comes in, but you don't have to study about them here. So chromosome, in short, can I write chromosome is DNA plus protein? Can I write that? DNA plus protein, and it is super condensed. Super condensed. Is it clear? Is it clear, everyone? Iram, Mariam, Rabia?
Yes, sir.
Now, how many chromosomes are there in each and every cell of a human? How many chromosomes are present in every cell of a human? Come on. This is basic. And you know the answer. 44 chromosomes. Right? 23 pairs. Out of which 22 pairs.
46 chromosome, 44.
Sorry. My bad. See, even the rest of us. 46 chromosomes. I'm so sorry. 22 pairs are called autosomes. and one pair, 23 into 2, 46 it should be. So bad with maths. One pair is called allosome or sex chromosome. The whole reproductive system was based on this, X and Y, right?
Yes.
X and Y. So if every cell of a human body has 46 chromosomes, how many DNA threads human body has in every cell? Same, 46. Because each thread will coil to become one chromosome. Does that make sense, Iram, Mariam, and Rabia?
Yes, sir.
So DNA is a very long molecule. It is condensed to become a chromosome. So 46 chromosomes, which means 46 DNA threads are present in each nucleus of a diploid human cell. If I ask the same question, how many DNA threads are present in the nucleus of a sperm or a ovum? What will you say? Do you understand the question?
Can you repeat the question?
The question is, how many DNA threads are present in the nucleus of a human gamete, sperm and ovum? It's 23, half the number which is present in our normal cells. Gametes are haploid, all our body cells are diploid. So diploid means 2 copies of genome, which means 46 chromosomes. Haploid means 1 copy, which is 23 chromosomes. So 23 chromosomes means 23 threads of DNA in the nucleus, which coil to form the chromosome. Do you understand that?
Yes, sir.
Very good. Perfect. And RNA is something that is formed from DNA by a process called transcription. In transcription, RNA is made from a DNA, and this RNA comes out of the nucleus. DNA never comes out of the nucleus except for the time when the nucleus dissolves and all the DNA comes out during cell division. DNA only comes out of the nucleus during cell division. All the time, DNA stays safe inside the nucleus, and any information it has to convey, it conveys through a minister, which is RNA. So RNA is formed from DNA. Genes, like Iram said, is nothing but a segment of DNA. Now we will read in the next class, not next class, but later on. Then what is special about this segment? Can all the segments of DNA be called a gene? Yes or no? And if no, then which special segments are called gene? And if yes, why all segments are called gene? That we will discuss later on. And chromosome is nothing but a super coiled, not just coiled, a super coiled form of a long DNA molecule present inside the nucleus of a cell. Are all these concepts clear to you?
Sir,
Yes.
I have a
yes.
question.
Yes, please.
Sir, how long is the DNA?
Yes. So it depends. All these 46 threads which are there in a diploid cell are not of the same length. Okay. That is why all chromosomes also are not of the same size or same length. Remember I told you Y chromosome is the smallest chromosome, right? So it is composed of a smaller thread. Total, if we take all the 46 chromosomes and we open it into a DNA form, all of them, we unwind all the threads and we keep all the threads one at the end of the other, one at the end of the other. The total length from one single cell will be 1.2 meters. Meter, around 120 centimeters. And how big is a cell? How big is a human cell? Which is the biggest human cell? Apart, like, which is a diploid cell, not a gamete. RBC, right? We can easily see RBCs under microscope. Okay. And RBCs are around, red blood cells are around 7 micrometer. How small is micro? It is 7 into 10 raised to power minus 6 meter. This is how long a RBC is. Okay. Now RBC does not have a nucleus. I hope you know that. If you don't know, just know it. A mature RBC lacks a nucleus. We have studied about this in 11th only. You know why a RBC does not have a nucleus, right? If not, please find out. I'll keep giving you some interesting homeworks. Find out why a mature RBC does not have a nucleus. But let's say when it was developing, then it had a nucleus. And in that nucleus, there were 46 chromosomes. And in the 7 micrometer cell, there was a 120 centimeter long DNA. It's bizarre, like, right? It is equivalent to say that inside your mouth, there is an elephant. Can you hold an elephant in your mouth? No, right?
No.
But if the elephant is a thread, then you can just coil it, super coil it, and keep it in your mouth. That's how inside the nucleus, the DNA stays. And why these molecules are so long? Because they carry information about everything of our body, about our eye color, hair color, this, that, genes, disorders, everything, all our genetics, all our genome. Okay. So
So,
that's how big. Yes,
so there are billions of cells.
there are trillions of cells, not billions.
Yeah, trillions of cells in the body. So if we combine all of
Yes.
their DNA, how long will it be?
You will surpass our solar system. From the sun, you can go easily, I think even more than that. You can go till, I think you can easily go till Pluto. If you just take one human body, take all the DNA out, keep it one after the other. Yeah. Trillions into 100 of kilometers. So trillions and trillions of kilometers. So I think you can easily surpass solar system. Or you can wrap Earth multiple times with just one human's DNA easily. You can gift wrap Earth. But the problem is, why can't we do that? Is because it is very tiny a molecule. It is very thin. It is so thin that you can't see it. So along with being long, it is super thin, super thin. So even if you hold one cell in your hand, you will not even, you can't even see the DNA inside. You have to pull the DNA from millions and millions of cells to see it under any circumstances. So single DNA can't be seen in this form, in its double helical structure. Biology is bizarre, but it's amazingly interesting. Okay, so today let's end at this clarity of these concepts: DNA, RNA, genes, and chromosome, because these are the concepts we are going to utilize for the rest of 3 chapters, 5, 6, and 7. Okay.